you, Sue, and thank you, organizers, for having me over. So the next 25 minutes or so, what I want to do is just, I was going through the agenda and just try and bring all the elements of this conference together. I've been involved with this conference now for the last almost 7-8 years. And it's an enjoyable conference, and I always find myself learning something new. I put this talk together with an eye to an immediate future, say the next 7, 8, 9 years. But before we go on, I wanted to, take a look back, right? So since about 1980, since Genentech's Humulin, biologics, have really emerged as a fantastic therapeutic class. Yeah, they're diverse, all the way from small peptides now to whole cells as drugs. If you look at, how these drugs are made, from E. coli to CHO proteins, then you've got the first real drug delivery technology in a pegylated protein. Now there are about 20 products out there that are being approved that are PEG proteins. The first antibody put, as a therapeutic, Rituxan qualifies as that. The first ADC, Mylotarg, it was introduced in the market, then withdrawn. Now it's, I think, back in the market, but, Kadcyla would be, in 2013, got its first approval for breast cancer. Enbrel is another drug where, a fusion protein on sort of a non-native sequence, and that's a seminal moment where it got, and it's a very successful drug to this day. And then you've got cells, the CAR T coming to bear, and multi-specific therapies, right? So what lies in the future? Perhaps mRNA technologies. I've highlighted, in my view, over the next 10 years, antibody-based medicine is going to be the mainstay of medicine. What do I mean by that? As therapies drawn from antibodies would probably reach the widest patient populations as new therapies, yeah? They are a tremendously successful therapeutic class too. So if you look at, today, this is from Nature Reviews in Drug Discovery from 2 months ago, 7 of the top 10 drugs are biologics, yeah? If you look at new therapies being approved by the FDA, consistently, about 30% of all new therapies that have been approved by the FDAs have been biologics, right? And so probably this reflects R&D portfolios that are about 7 to 10 years old, yeah? But I haven't done the systematic study, but if I venture to, compute that about 50% of, R&D portfolios today are MAB or MAB-based therapies of companies. And so what you see as 30% today, perhaps 7-8 years from now, it'll be 50% of new therapies being MAB-based, right? MABs, ADCs, single chain antibodies, fusion proteins, and the like. But ever since, we've learned how to, or we are still learning how to make antibodies and antibody-based medicine, we put this data together based on, approvals. So within the biologic space, clearly antibodies dominate, right? So antibodies, monoclonal antibodies are the dominant therapeutic modality. And then if you look at what a patient sees, right, ultimately, either through a caregiver or a patient having to administer the drug to themselves, right, then there's an increasing pressure for producing liquid dosage forms because they're more versatile, and then into more competitive dosage forms, yeah? Going from IV therapy to subcutaneous therapy. And that trend is only going to increase. I mean, even if you take cancer as a wide 100 diseases, right? Some of them we might find disruptive cures, but realistically, the disease, our goal would be, or the realistic goal is to convert it to a chronic disease. That's something that you can manage. And many cancers today, you can do that with awesome therapies, right? And even in those spaces, right, there's an increasing pressure or increasing need from patients to have more convenient dosage forms, right? So antibodies I view them as the great equalizer. Why? Because, okay, we appreciate their drug-like properties. They have amazing specificity. They are a lot safer, and you can, reduce the frequency of dosing. But the second bullet, I think, is what, in my view, really brings that therapy across to be applicable to a wider patient population, right? Because how you produce these molecules, there's a certain, conversions of technologies that enables you to produce them, right? You get predictable performance today, right? And as an industry, we're sort of moving to incrementally, improve that performance. And what that has done is enabled the translation of an idea to a therapeutic. And that has lowered the barrier to entry, right? So if you imagine, in the pre-monoclonal antibody days, you would require boutique purification processes, and a tremendous amount of expertise. Not that you don't need that for antibody production or delivery, right? But it sort of has lowered that barrier for smaller companies to enter into the game, right? Now, it's interesting, in 2003, when I was moving from Pfizer to Amgen, Pfizer had evolved itself from a classical pharma company to a biopharma company, right? And today, if you look at companies around us, most companies are hybrid pharmaceutical, biopharmaceutical companies, yeah? Already more than, I would say, 80 unique products being approved by the agency. And there's a really nice review that came out at the end of December in Nature Biotech that really summarized all the antibody-based therapies or biologics that have been approved. And now you've got biosimilars that are being approved. But antibody therapies are also evolving, right? So you've got antibody drug conjugates, not only cytotoxic. There's talk of loading up antibodies with other types of small molecule drugs, right? You've got multi-specific antibodies. Hemlibra is a fantastic example of an approved drug. Last year, you may have read, there was a really nice paper that came out of Sanofi of a tri-specific antibody for HIV therapy. Yeah. Then, one of the drawbacks or one of the limitations for antibody therapy is that getting entry into cells can be limited. And now you have VHH antibodies. They're really small, some of them from camelid origins, also approved in the market, Fabs or fusion proteins, like Enbrel or Enbrel-like molecules. And really, there could be a variety of different reasons why you'd want to do that. Some commercial, some, very technical as to why you may want to do so. But as you're developing any of these therapies, an amazing thing happens, right? From research to production, many a times you start with microgram quantities of molecules, and over the next 7 to 10 years, or sometimes 5 to 7 years, learn to make tons of drugs, right? And through that process, in my view, what would be a disruptive sort of competitive advantage is right at that, what I've marked out, that interface between research and when you enter clinical trials, is not only now do you need a molecule that is biologically active, but that molecule needs to have druggable properties. It could be a great binder, but if it's not druggable, then it's just going to take that much more time to develop it, right? The kinds of words we use through that development, and then there are themes, there are thematic talks all through the sessions with the next week that talk about, molecular design, high throughput screening, or developability in that early space, right? And then the game changes once you enter phase one, is you want to know is this drug safe, and you want to do so expediently with platform type approaches. And then when you go to phase 2B or phase 3, that's when we start using words like well-characterized, competitive dosage forms, and strive to have consistent processes. So I want to, I personally view developability as a key enabler to the drug development process as it applies to, protein and antibody-based therapies. You can view, the entire problem in sort of 4 stages, right? Once you design your molecule and once you've chosen your molecule, that's it. You get what you get, and don't be upset, because after that, it's the next 5 years or 7 to you have to develop that molecule with all its liabilities and advantages, right, inherent. And the companies or organizations that figure out how to affect molecular design have effective screening tools, right? And I view that, and I use that word effective screening tools not lightly, because every burden that you put in, or every selection criteria that you put in to an already difficult task of having a high binder or a biologically active molecule, is a deterrent, right? Is an additional step the entire organization or the machinery has to take. So what works is important, what is predictive is important to find, and equally importantly is what is not important, right? And that's very important to know. That has to, bear down on your scale-down models, and then ultimately, at your at-scale performance. And I've just given examples of issues, right? There's enough talk about high viscosity. You can have issues of reduction. You can have poor chemical stability, and that can that most often than not, limit shelf lives of drugs. Or another area that, again, in the next 7, we don't fully understand yet, or how to leverage it, is, why are some antibodies, subcutaneously 50% bioavailable, while certain antibodies are 80% bioavailable. Now, if all other things kept equal, right, if you can make a highly bioavailable antibody, then it just simply means that you have to produce far lesser of it, right? It just, of course, it's a simplistic argument, but there's an argument to be made there, right? Having said that, there's another transformation that's underway, and it involves all of us, because all of us are patients at one point or the other. if we assume that 30% of new modalities, new therapies today are going to be biologics, and in the next 7 to 8 years, or the next decade, they're going to be 50%, then how we take medicine is changing, is fundamentally changing, right? Humans have been taking medicine since the beginning of time, since the beginning of recorded history, right? And all of that medicine has been taken orally. Till about 1950, where you have a formal IV therapy. It was given before that also, but just formally. But from 1950 to 1980, you've got about 10% of all new medicine, right? For every, in the case of NCEs, for every 10 NCEs, right, you just had one parenteral NCE, right? And so you had 10% of medicine. And since then, there's a steady increase and there's a steady rise, right? And so imagine now that that world is here and now, where we have to, adapt fairly rapidly as to how are we going to, what are our therapies going to look like to a patient, right? They have to be more convenient to ensure, compliance, to ensure adherence to therapy, because, generally, all of us as patients, at some point or the other, we don't like to be reminded of our disease. And the first thing that occurs is non-compliance, right? And that's a very real problem that we'd have to solve. So, increasingly when it comes to biologics, the better you integrate all the way from lead candidate selection and have a view of what's going to happen 10 years from now, right? The better that view, the better your product's going to be, right? The better our product's going to be. And that spans, all the way from lead candidate selection to formulation to drug substance processes, drug product processes, and how they integrate into devices, right? And that is going to be so key. From a patient's perspective, right, a pre-filled syringe is a pre-filled syringe, right? But if you look at from a developer's perspective, these are all the things that could go wrong with the pre-filled syringe, and at some point have gone wrong, right? Have led to major product recalls. But that has to be integrated into the entire development process, right? So research chemists have to really have the view that, our fates are tied together because it's ultimately what makes it to the patient and not a very active molecule, or just not make enough of the drug substance and I'm done. Or the drug product guys, my product's stable, who cares about integrating it into a device. And that is not going to serve anybody well. But working together and breaking those silos, companies to do that better are likely to succeed. Again, that's my view on it. Gene and cell therapies are emerging, right? You already have products that have been approved. Yeah, you've got ex vivo approaches and in vivo approaches. Yeah. Exciting therapies, they will, in cases of where there's a single gene to be modified, clearly present opportunities. But the challenges with these therapies is going to be how are we going to make this therapy available to wide, large patient populations, right? So we'd have to understand disease biologies better. We'd have to understand the complexities and of manufacturing processes better, the scalabilities of these technologies better, right? Clearly, they're very successful, breakthrough, transformative technologies, but what's the next thing is going to be, when you have 1 million patients, when you have 5 million patients? For certain kinds of diseases, how are we going to make these therapies available? That's an open question. Viral versus non-viral gene therapy, right? Exciting area. With viral-based approaches, you've got a ready delivery package, but limited in the amount of genetic material you can pack in. Questions about can you have repeated dosing with non-viral gene therapy? Yeah, you don't need a bioreactor, but then questions of what is the efficiency? Are you going to get it to where you want to get it? And those issues start coming up. And there's exciting progress, in that area. And what are the barriers to deliver these technologies, right? You've got the good old, the in vitro stability problem. But then you also have what happens, when your formulations start interacting with blood components. Okay, you've solved that problem. How do you get that therapy to where you want it to be, right? Do you want systemic exposure? Do you want, a particular target organ in mind? Now, certain therapies lend them to certain target organs easier than others. But again, if we're serious about that, this problem is going to have to be solved. Okay, once you've delivered payload into a cell, how do you get it out of the endosome and make it available? And then if your genetic material is DNA, then how do you get it to the nucleus, right? So again, this is, I've broken that problem statement down, and progress needs to be made in each one of these areas, and is being made, right? It's fairly exciting area to be in. So I'm 3 minutes out, and I guess I'd like to, summarize my talk. Is biologics, are the new, kind of medicine. It's no longer new. It's here to stay. But it represents a very diverse set of modalities, from small tides to whole cells, right? But as I said, map-based therapies in the shorter term, in the next 10 years or so, I think would be the dominant therapeutic modality. The broad success of DNA, RNA-based technologies will hinge upon, understanding biology, the big delivery challenges, and then the scalability and manufacturing complexity. And in all of these, regardless of the modality that we are talking about, whether it's a peptide or a cell or antibodies, today they necessitate parenteral administration. And this is a transformational change that we are going through as a industry or even as a society, and how we design those dosage forms, right? How that medicine looks to a patient, right, is going to be very, very important going forward, simply because, at the end of the day, if a patient's not, for whatever reason, and good reason per se from the patient's perspective, not adhere, not comply to a therapy, then all that effort, could potentially be in vain. So with that, I'll, end my talk, and, thank you for your attention, and, I'd be happy to answer any questions that you may have. I believe we have some time now, and then there's going to be, Tony and I are going to make myself available at 5:15 at a booth. So thank you for your attention. Yeah.